much. I'd like to thank the organizers and the program committee members for inviting me to Boston and also giving me opportunity to talk about our recent study. So today's title of my talk is Modeling and Simulation of Neuronal Morphology. So there is a basic hypothesis here. So we consider that morphology and structured information representation in the brain, such to read out the genomic blueprint, and also information provided by the environment. So to know how neurons obtain their morphology and how neural circuits form their patterns is to know how the brain acquires its information basis during development. But such morphology and structure formations are actually performed by molecules and processed in stochastic environments. So we need some mathematical tools or computer simulations in order to explore such kind of mechanism. So during neuronal development, a certain number of neuroids are produced around the soma, but even in a uniform environment, one of the neuroids comes to be elongated eventually, and it becomes an axon. This process is called polarization. And after that, the polarized axon will guide some molecules, say diffusing molecules, coming from distant places. And this process is called axon guidance. After that, synaptic connections are produced. So these processes are in total called structural plasticity. So in this talk, I address in particular polarization and axon guidance. So such structural plasticity is very interesting, I think, because it is a decoding process from analog molecular signals to digital morphology. And also, molecular signals could be very weak in comparison to the noise that should exist in the molecular world. And also, structural plasticity can be symmetry breaking phenomena induced by noise. So we want to know the mechanisms underlying this kind of process. So we believe that even single neurons or even single subcellular structure of neurons can do process information provided from the extracellular world. So the first topic is spontaneous neuronal polarization. So between stages 2 and 3 during neural development, one of the neuroids is selected as an axon candidate. So this is a typical symmetry breaking phenomenon because you know that a typical pyramidal neuron has only one axon and multiple neuroids, dendrites. So this process is called polarization, and it has several interesting characteristics. One is spontaneity. That is, a neuron is spontaneously polarized even in a uniform extracellular condition. The second one is stabilization. That is, once a single axon is selected, remaining neuroids cannot be elongated in most cases. So this is very important for stabilizing the symmetry breaking of the system. The third one is correction. Sometimes multiple neuroids mistakenly happen to be selected, but such a failed pattern is flexibly cancelled out to yield a favorable pattern with a single axon. So the question here is, what is the mechanism of such a flexible neuronal morphogenesis? to answer these questions, we have developed a mathematical model here. It is a compartment model consisting of a single soma and several neuroids. And also we assume axon determination molecule, which is signified by factor X here. The model is represented by these simultaneous differential equations. One is an ODE, representing the concentration of this factor in the soma, and the others are spatial temporal PDEs, representing the concentration, each representing the concentration along each neuroid. So in these equations, we have several terms. So this represents gene expression in the soma. And as a natural assumption, this molecule is degraded or inactivated as the time proceeds. And it diffuses, represented by these three terms. And also we assume that this molecule is actively transported from the soma to each neuroid tip. And also there is a decoding molecule. Actually, it is assumed to be a cytoskeleton regulating molecule, and it is represented by factor Y here. So it works at each neuroid tip. Actually, it is called growth cone, and it is activated by the axon determination factor X, that is X, and also it shows a bistable hysteresis like behavior against factor X concentration like this. So in other words, this molecule works as a molecular switch. Although we do not specify the molecular species of factor Y, for example, H-gas is known to behave like a molecular switch, being highly activated in the neuroid, which has been selected as an axon candidate. So we did some mathematical analysis. So these are actually the analytic solutions. So first, we see that it is difficult for factor X to diffuse back to the soma if the neuroid length becomes long. So this in this panel, the horizontal axis denotes the neuroid length, and the vertical axis denotes the accumulation percentage of this molecule in the growth cone. So that is, the longer the neuroid becomes, the more this molecule accumulates in the growth cone. That is, this molecule works as a local activator. So, in other words, this molecule cannot return back to the soma if the neuroid becomes long. So if there are several long neuroids, so the somatic concentration would decrease because the consumption in the neuroids may be larger than the production in the soma. That is, this structure is similar to the global inhibition. Mathematically, there is a structure like local activation and global inhibition. The system may exhibit the winner-take-all mechanisms. So such a mathematical analysis, this is mathematics anyway, so but such kind of analysis can shed light on the mechanisms underlying flexible neuronal polarization. So as the time proceeds, the somatic concentration of this molecule increases. Then in this case, the factor X concentration in one neuroid may happen to arrive at this threshold. Then this neuroid becomes a winner. Then it elongates. But after that, the somatic concentration would decrease because of the decrease in the diffusion back. So the active transport tension is proportional to the somatic concentration. This character would suppress the other neurons to become an axon candidate anymore. So this is a mechanism of spontaneity and stability of this system. Okay. Yes, once a single neuron elongates, other neurons cannot be a winner anymore. So another interesting character of this system is correction. So if the factor's concentration arrive at this threshold in 2 or more neurites, they become winners and they elongate. But in this case, the somatic concentration would decrease too much because there are 2 or more long neurites. In this case, the active transportation amount would decrease so much. Then the factor X concentration of the winner neurites may arrive at the lower threshold of this hysteresis curve. Then those neurites change into losers again. Then the somatic concentration would increase again, which encourages other neurites to become an axon candidate. So this is a mechanism of the error correction of this system. So this one is some sort of mathematical analysis. But, okay. On the other hand, our collaborator, Dr. Inagaki, previously found a molecule whose name is shooting 1, is highly expressed in the growth cone during neuronal polarization. So this molecule is actually transported from the soma to the neural tip. So this is active transportation, and the neuron may be elongated like this and may shrink. But such local movement is highly correlated with the concentration of this molecule in the growth cone. So actually, the red line shows a smooth velocity of the neural tip, and the green color shows the GFP intensity of this molecule in the growth cone. So you can see some nice correlation between these two factors. They also did a Kyoto experiment. So before UV irradiation, Kyoto shooting 1 highly expressed in the growth cone. But after UV irradiation, the green color changed into red one. But the red color decays as the time proceeds because of the diffusion of this molecule around the neurite. From this experiment, we can measure the decay time constants for a number of neurites. And then, based on the linear relationship between the neurite length and the decay time constant, we can estimate the diffusion constant. So from this experiment, we can estimate the parameter value of the model. But in order to simulate axon elongation process, we need a downstream model of this molecule, but it is completely unknown. So we need a model here. In our model, we employ the elastic model of neurite. It's an elastic spring, and its elastic force is given as by a nonlinear function of this molecule's amount. Well, because this model has several parameters, they are estimated by means of the least square method as to incorporate the experimental data. And this figure shows the reproducibility of the model. So each red point denotes a single data point corresponding to a single neurite in the experiment, and each blue point denotes a model's prediction for the same neurite. So as you can see, some sort of nonlinear character of the downstream can be represented somehow by the model. Okay, so because this model, the parameters are determined based on the experiment, it can be evaluated in a quantitative manner. So the right panel shows computer simulation. So as the time proceeds, the neural length of the axon candidate becomes long, accompanied by the concentration increase of this molecule, the corresponding growth cone. The left panel shows the experiment. So similar to this one, the neural length of the candidate axon becomes long, accompanied by the concentration increase of this molecule in the corresponding growth cone. So we can say that the symmetry breaking phenomenon between neuroids can be reproduced by a model in a quantitative manner. Okay, the second topic is a high signal-to-noise ratio in chemotaxis. So chemotaxis is a property, probably all of you know, it's a property that a cell detects the gradient of chemical substance and moves up toward its direction. So it is closely related to the axon guidance system, we believe. So important is usually the gradient of chemical substance is so gentle that the cell should detect very weak signal even in very noisy environment because the molecular world is very noisy. So we try to understand the general principle underlying this process. So in chemotactic cell, experimental study has found some spontaneous activities below the membrane. So in this movie, transient increases in PAP3 concentration is actually observed, and seemingly they induce lateral extensions to that direction. So intuitively, such spontaneous activities of the signaling molecule could be the key for understanding the cellular mechanism of sensing weak signal provided from the environment. In order to know the mechanism, we developed a biochemical model. So this one is the PAP signaling pathways, simplified version. And PAP3 is activated by kinase PI3K and inhibited by phosphatase P10. And the dynamics here is given by this equation for phosphorylation and for dephosphorylation. And all terms are actually represented by Michaelis-Menten type equations. So, okay, this one is a simplified version, but still four-dimensional dynamical equation. But if we apply some sort of assumptions, such as positive feedback is fast enough, we can reduce this system into a two-dimensional one, governed by the PAP3 concentration and the P10 concentration provided by the negative feedback. It is represented by W here. Then we applied an analysis on the two-dimensional phase plane consisting of the PAP3 concentration x-axis and the P10 concentration y-axis. So we found that this dynamics has a similar structure to the well-known Fitzhugh-Nagumo neuron model. That is, there is a fixed point here, but if the system happens to go over these separate weeks or nuclein, it emits a spike-like activity and then go back to the fixed point again. So this is the mechanism of the PAP3 spike generation of the system. So the PAP3 system is an excitable system. So such a spike-like activity is actually generated by the fluctuation of the system. So we evaluated the potential field produced by the PIP3 signaling pathway, and also we evaluated the spike occurrence rate. That is a transition rate from this state to this state. So like this. So we evaluated this transition rate like this. So here, this transition corresponds to the first passage event reaching this unstable potential barrier, potential peak. And the important is transition occurrence rate is given as exponentially to the potential barrier height. So this is exponential modification. So a theoretical consideration, this kind of theoretical consideration would lead to the mechanism of the signal amplification done by this system. When there is a gradient of chemical substance like this, it's quite gentle, such as 5%. But the existing study has clarified the localization of the signaling molecules. P10 will accumulate at the cellular side of higher concentration, this side, and P10 will accumulate at the opposite side. Even if the intracellular gradient of these two kinds of molecules is linear to the concentration of the extracellular chemoattractant, such kind of linear modification will change the potential barrier height in a nonlinear manner. And also, the spike occurrence rate is exponential to the potential barrier height. This is a nonlinear amplification of the signal provided from the extracellular world. So this one is actually the mathematical analysis. But in order to confirm this scenario, we did a Monte-Carlo simulation. This is a more detailed signaling pathway of the PIP3 signals. Actually, you can observe PIP3 signal spikes are produced highly frequently at the cellular side of a higher concentration of chemoattractant. But such kind of activities rarely occur at the opposite side. That is a lower concentration of the chemoattractant. So we consider that the PIP3 system is a nonlinear system so that spike activities are spontaneously produced. And then such kind of spontaneous activities are actually used for nonlinearly magnifying the signal provided from the extracellular world. So this one is, we believe, what the chemotactic system does, but the essential part can be shared by the growth cone guidance system. So the next part is about our ongoing project about bidirectional responses by growth cones. So this is a growth cone. So, which is the machinery system responsible for neuron wiring. So even in this small structure of neurons can do some information processing, and one of them is bidirectional responses to the gradient of same molecules. So in this experiment, the receptor modification can change the turning direction of growth cones. So in this case, it shows attraction to the chemoattractant, but in this case, it shows opposite behaviors. So such kind of bidirectional responses are thought to be important for performing situation-dependent Axon guidance during the developmental stages. So our collaborator, Dr. Horn's group, found that growth cones show bidirectional responses to the net neuron gradient when changing the ratio of cyclic AMP to CGMP basal pride. So this figure shows that when the ratio is large, the growth cone shows attraction, but if the ratio is small, the growth cone shows repulsion. So this phenomena would probably due to the neutrino-induced calcium signal, which is under the balance between the CGMP activation and the CGMP inhibition. So they also found that when the semaphore gradient is applied, the calcium level is elevated more in the near side than in the far side, but the elevation level is larger in the attractive condition than in the repulsive condition. So we can consider that gradient direction is somehow encoded into the steepness or basal concentration of the intracellular calcium. But the question here is how gradient information is encoded into calcium signal and how calcium signal is decoded by its downstream. So in order to answer these questions, we made a model. So under the observation that in this growth containing system, there are two kinds of balancing factors between activator and inhibitor. So this is a simplified signaling pathway under the neuronal signal. So this is calcium. In the upstream of calcium, there is a balance between CGMP activator and CGMP inhibitor. And also in the downstream, there is another balance between the CAMK2 activator and the calcium PP1 inhibitor. So we developed a model that is a combined activator-inhibitor model integrating the upstream model and the downstream model, both represented by reaction diffusion equation like this. So actually the model itself is a complicated one, but we can apply a Taylor expansion, that is a linearization to this model along the growth cone coordinate. The situation is like this, the gradient is like this, and the growth cone coordinate is like this. So we can obtain a very simplified mathematical equation like this. So this one shows the decoder's difference between the near side and the far side. And we assume that if this value is positive, the growth cone will show attraction. And if this value is negative, the growth cone will show repulsion. And interestingly, this equation is actually dominated by two ratio values. The one is dependent on the equilibrium concentrations of activator and inhibitor, and the second one is the concentration difference of activator and inhibitor between the two sides. That is the near side and the far side. But this kind of values can be evaluated experimentally, so we can predict in which condition the growth cone will show attraction or repulsion. So this one is the previous experimental result. So if we change the ratio of cyclic nucleotides, the growth cone may show attraction here and repulsion here. So this one is a solution of the model. And this one is a ratio of cyclic nucleotide, similar to this one. And in this region, the growth cone will show attraction, and in this region, it will show repulsion. And it shows a nice agreement with the experiment. However, if the activator's response, either of the activator response or inhibitor's response, may show a sigmoidal nonlinearity like this or like this. The ratio behaves like this, so they produce two crossing points. In this case, the growth cone may show more complicated behaviors. So in this case, the growth cone may show repulsion here, attraction here, and repulsion here. So it's a more complicated behaviors. But such a complicated behavior is actually observed in the real growth cone. So this is another experiment. The mixture of cyclic OMP and cyclic GMP is directly applied by a pipette, and they will produce some intercellular gradient like this. That is a higher concentration in the near side and the lower concentration in the opposite side. But in this case, if we change the total concentration of this mixture, the model predicts that the growth cone will show repulsion in this region, attraction here, and repulsion here. But this behavior is actually observed experimentally. So this is the ratio is actually fixed. The total concentration is changed. But in this case, the growth cone shows repulsion corresponding to here. This region, the growth cone shows attraction corresponding to here. And here it shows repulsion. So such a very complicated behaviors of growth cone turning is predicted by the model and then experimentally confirmed. Okay, so this is a model study. In the next part, I would like to introduce wide-scale simulation studies of neural systems currently done in Japan. So as Dr. Kawata mentioned, Japanese government and Riken are now constructing a supercomputer whose name is K, and it achieved the world's fastest impact performance of 8.162 petaflops in the last June. So, this name K comes from the Japanese word meaning 10 peta, which is expected full performance of the computer. So this one is not computer graphics, it's a real picture of the K supercomputer. Okay, so this supercomputer is actually located in Kobe area, close to the venue of the last Neuroinformatics meeting. So after construction, it will have more than 80,000 processors, each with 8 cores inside. So the total number of cores will be 640,000. It's a huge system. So what is the purpose? There are a couple of application projects ongoing. One is for nanoscience, and the other is for life science. The life science application project is called Next Generation Integrated Simulation of Living Matter. To promote research and development of simulation software which helps understand phenomena, biological phenomena from molecules to the entire organisms. So within this project, we have a team to develop a software for brain and neural systems. So we are now developing neuron circuit simulators. NeuroMorphKit is a software platform for simulating neuronal morphogenesis. And circuit simulator Nest is a simulation tool to simulate and predict the signal processing for even 10 million neurons in the cortex. So this is the situation of implementation of Nest on the K supercomputer. The high degree of polarization is actually achieved by using the architecture, hybrid architecture of NPR and OpenMP threads with more than 8,000 cores on the K supercomputers. So it shows a very good scaling up to 4,096 cores, which is signified by this number. This is the efficiency, and actually one is ideal. So 0.77 is quite good. And also it shows good scaling for more than 8,000 cores, which is signified by this number. But still there is some issues, communication computation balance, so they are trying to improve this board. So we also try to simulate whole brain level circuits. One software targets a visual system consisting of the cortex, retina, eye optics, and eye motion plants. So this software is actually developed by Dr. Usui, Dr. Kenji Dora, and Dr. Shin Ishii. And the other one is a whole brain simulator for the insects olfactory system, which is done by Dr. Kanzaki's group. So this is a simulation study of whole olfactory system of insects. So we try to make a circuit model of detailed morphological neurons and to simulate odor orientation behaviors of silk moths. When facing the odor source, the moths generate exploratory behaviors, and such kind of behaviors are actually driven by LAA, VPC, premotor center area located in the brain, insect's brain here. So based on the imaging and physiological experiment, we estimated the network parameter like synaptic connection between neurons. Then we developed a circuit model consisting of multi-compartment Hodgkin-Huxley type neurons, and it is implemented on the neuron simulator platform and also implemented on the K-supercomputer. Surprisingly, this software simulator shows a very good scaling, almost linear, up to 10,000 cores on K-supercomputer. Okay, so the last part is cytoskeleton-based morphogenesis. This is a simulation study. So in the early stage of neuronal development, actin and microtubing are localized and, at the neuronal tips and along shafts. But in the later stage, actin and tubing are localized as synapses and along dendritic shafts. So because neuronal tips and synapses are the most important sites of structure plasticity, we consider that cytoskeleton, especially actin, is crucially involved in structure plasticity. So we are interested in cellular morphogenesis. But cellular morphogenesis is a typical multi-physics system. So it is regulated by intracellular signaling, represented by reaction diffusion of the key molecules. For example, this movie shows dynamic cellular shapes driven by its molecule whose name is CDC42. And also, another important player is cytoskeleton, of course. It is polymerized, depolymerized, branching, and some others. And this maybe shows actin dynamics within the growth cone. Okay, so you can see that cytoskeleton is very important in the growth cone movement. And also the cytoskeleton affects the membrane. So if actin polymerization is inhibited, the complicated shape of cell changing to a round one, suggesting that the cytoskeleton, in this case polymerized actin, mechanically sustains the membrane. And also, okay, so the membrane depends on the cytoskeleton, and it also defines the boundary conditions for reaction diffusion of intracellular signaling. So this is a multi-physics system consisting of at least intracellular signaling, cytoskeleton, and membrane. So we try to simulate this system. So for this purpose, we need a model of actin filament. So actin filament is a polymer. It is polymerized and depolymerized, and also its treadmill movement would provide driving force for cell motility. So this is called actin treadmill hypothesis. And also, there are many events in the actin system. If a molecule called Act2/3 complex is attached to the tip of the filament, it becomes a seed of filamentous branching. If a capping protein is attached to the tip, the polymerization is terminated. And another kind of event is seedling, which is regulated by a molecule named coffin. So these processes are modeled in order to simulate actin kinetics. So in our system, there are three major elements: intercellular signaling, cytoskeleton, and membrane. So individual biophysics are solved within each time step in the computer simulation. So in the simulation, we use the compartment model. That is, the space is represented by two-dimensional compartments. The membrane is expressed by compartmentalized polygons. And also, actin filament is expressed by line segments. So more concretely, we first solve reaction diffusion equations for intercellular signaling in order to know the concentration of key molecules. And then we reorganize actin filaments by performing actin kinetic simulations in a stochastic manner in the level of a single event. And then we have array of actin filament. Then we solve the energy-based membrane deformation problem whose energy function is dependent on the array of actin filament. So this is Outgrowth. So this is the simulation result of self-organization of lamidopodia. So in this figure, each red line denotes actin filament, single actin filament, and the green point denotes a single active capping protein. So even in this simple simulation, we can reproduce several biological findings. For example, Act2/3 is activated in the vicinity of the membrane, and in the simulation, this is a simulation result. So as you can see, the concentration of this molecule is high in the vicinity of the membrane. So by introducing the actin treadmill hypothesis, we can simulate chemotactic migration. When there is a chemotactic gradient like this, the cell likely moves up towards the gradient. Yeah. Yes, this is a simulation. So the cell actually performs chemotactic migration by dynamically reorganizing the actin filament. This is another simulation. This is a cellular invasive migration. Similar to the previous one, there is a chemotactic gradient like this. And also there are some obstacles which correspond to other cells or extracellular matrix. So in this case, the cell invades the narrow slit by dynamically changing its shape driven by dynamic cytoskeleton. Yes, this is some sort of simulator, and it works on the case of a computer also. It is tested up to 512 cores, it's not so large. And under the hybrid of MPI and OpenMP, and it accomplished moderate polarization about some membrane energy optimization, but a total polarization is not very sufficient So we need to improve the algorithm about actin kinetics in order to achieve further efficiency on the K-supercomputer, which is a remaining issue. But in neurons, not only lamellipodia, but also filopodia are very important. In order to simulate filopodia, we need some model about bendable actin filament, because filopodium is a bundle of actin filament and which are linked by a linker protein fascin. So in this in vitro experiment, filopodia-like structures appears in a membrane-free environment, no membrane, only with actin, AP2/3, and fascin. So by changing the concentration of AP2/3 and fascin, the actin system produces various kind of structures, including star-like structures. So we try to reproduce such kind of filopodia-like structures. So this is a preliminary version of the simulation, and actually star-like structure of filopodia is self-organized by introducing the model of Bendable F-action and linker proteins to the previous multi-physics system, incorporating the actin system. So this is the last slide. So the current our study is modeling. So as Dr. Kawato mentioned, we have started a new project about brain-machine interface in daily living. So which is to know the brain's information processing in-house. But similar to this one, we want to know the growth cones information processing in vivo. So in order to know such kind of information processing, one possible way is growth cone machine interface, analog to brain machine interface in daily living. So this is a kind of joke, but not only modeling, but also decoding based on the model we have constructed might be an important issue. So we try to explore to that direction. So this is the end of my talk. Thank you very much.